Hello and welcome back to the Biology 211 lab. So the plan for lab 1.5 follows here. There will be questions on the quiz for week 2 from lab 1.5. Some of you have already done that. Others will be doing it soon, I'm sure. We will analyze the sequence of the GAPC gene, obtain the kale, the cabbage, and the Arabidopsis nucleotide sequences from the file on Blackboard.
We will also do a writing activity. This writing activity has 2 separate parts. In the first part, you will peer edit 2 different versions of a Biology 112, that is cell biology's, writing samples. One of these is called the short version and one is called a long version. You will use the rubrics associated with those 2 writing assignments to evaluate and provide a grade for those 2 assignments. And that is worth 5 points. The second part is to peer edit your teammates' lab reports. And I'll explain how we'll do that in a moment. But each of your introductions and figures and figure legends will be evaluated by 2 of your other teammates. And in doing so, you will use the rubric that is associated for the introduction assignment. This part of the writing activity is worth 15 points.
So what's due from this lab on Thursday at 11:59 p.m. is worksheet 1.5. plus the worksheet peer editing part 1 of the Biology 112 cell biology writing samples, and part 2 lab report. So here's the module 1 flow from your syllabus. But today what we're doing, it's not shown here, it must be hidden under the red line here, but we're analyzing the DNA sequences, plus we're doing the peer editing practices. So the first part is analyzing the DNA sequences. You'll go to this website here, and then you'll get the sequences from the kale, the cabbage, and the Arabidopsis sequences found on the Blackboard file. You copy the sequences from this Word document. So first you copy the kale sequence, and then you'll put it into this field here, and then you'll click this button here called Blast. And the data you get from that you'll use to complete worksheet 1.5. That is due, as all assignments for this week, on Thursday at 11:59 p.m. The second part of today's lab is a writing activity. Now this will take more time than the sequence analysis. Now, similar to what you found in Biology 112, there are very detailed rubrics for Biology 211's writing assignments. Each criteria of your introduction is on the left side of this rubric. The heading up here on the top indicates the amount of credit awarded for specific performances of each criteria. So to get 100%, you would want to make sure that you follow these criteria. If you're cool with 85%, then go ahead and follow these. When you're editing someone else's work though, what you'll want to do is, as you're, say, reading their title, which one of these fits the title that you're reading? And then just circle that for each of these criteria, such as shown here. But don't be afraid to mark something low, say here, or even here. Just because they're in your group doesn't mean that you shouldn't grade them fairly, because the goal is that their paper is going to become better and they will get a better grade. The last thing you want to do with somebody in your group is say, oh my gosh, that was the most beautiful thing I ever read, and then they get it back and it's a 70%. In fact, I can promise you that when I send my papers off for my colleagues and friends to evaluate, they are definitely not too kind to me. They are very free at ripping my writing apart. And I want them to, because I want my writing to get better. To determine the number of points, what you do is you circle the appropriate box in each row, calculate the points earned by multiplying the percent column, written as a decimal, to the possible points for that category shown here. 100% of 5 points is 5 points. 85% of 5 points, so 5 over here, is 0.85 times 5, or 4.25 points. Likewise here, 55% of 5 points here is 0.55 times 5, or 2.75 points. So this is how you will end up calculating the points that your teammates have earned on their paper. And you total it up in each row to get an earned score out of the possible points. As practice for Lab 1.5 Part 2, you're going to peer edit a Biology 112 cell biology writing sample. So you'll see the short version in Lab Manual on pages 47 through 48. Read each version a second time, circle errors, and make comments on each paper. Use the provided grading rubric to grade each version. Record the grade you assigned for each version. Complete worksheet, peer editing Part 1. It's worth 5 points, and that's due Thursday at 11:59 p.m. with your other assignments. Now that you've practiced peer evaluating the examples from Biology 112, I want you to work in your teams of 3 or 4. I'm not going to read this in great detail because this is just an example. You don't have to follow this exactly like this, particularly since most of your teams are teams of 4. But the main point I'm trying to illustrate here is that every introduction and the figures must be evaluated by 2 individuals. One per person's introduction is evaluated using the rubric found on pages 11 and 12 of your week 2 worksheets. But the point I'm trying to make here is that everybody's introduction must be peer edited by 2 members of their teams. One paper is given to one reviewer. That reviewer peer edits it using the rubric. They then pass that review onto a second reviewer, and then they will make their own comments on it. Now, since we're not face-to-face and we're online, those comments could be made on a Word document and just using different colored fonts. I write green and purple here, but it doesn't have to be those colors. And then in the end, the second reviewer will return it back to the original author. So using this plan, or a similar plan on the previous slide, swap papers with both team members and use the rubric on the draft itself, the original author's draft. Write your name as editor, write comments, and a score for each criteria listed and a total score. For each criteria and a total score. The second editor should make independent evaluations using a different colored ink, but on the same copy. They could even circle comments that they agree with. Altogether, I want you to score the introduction, the figures, and the figure legends on each criteria listed in the rubric. Peer editing assignments will be graded based on the effort made by the different editors and the making of a significant number of constructive comments on your peer drafts. After both reviewers have completed their evaluations, return the work to the authors, discuss the comments if you have time, and resolve questions. Remember that as you receive comments on your writings, don't take it personally. This really is an attempt to make your writing better and for your grade to improve. You have to turn in the peer editing worksheets with your Thursday assignments at 11:59 p.m. Looking ahead to next week, remember that on Tuesday, this is going to be a free lab day. Nothing specific for Tuesday of next week. This will be a day for you to work alone, to get your introductions done, your figures done, and your figure legends done. If, by chance, you have these already completed, then you can work on other assignments. Or you can just relax. Wednesday and Thursday of next week, we're going to begin our second lab module, and in this lab module, we'll be learning about C. elegans, which is a very powerful model organism for genetics, and we're going to be using it to understand behavioral genetics. All right, that's all I have for this video podcast. If you have any questions at all, please make sure you reach out to us. If not, we'll see you in lab or lecture. Bye.